All right, good morning, everybody, and welcome to the first talk of this year of the UAB Informatics Institute PowerTalk Informatics series. And I'm so happy to introduce today's speaker. I see many of you here that know him well, and so he doesn't need any introduction. One of the most prominent people right now in of all time in informatics. I am proud to call myself an alum of his program, so very proud to have had him as a professor and mentor. And our director of our Informatics Institute, Jim Cimino, unfortunately, he's not able to attend today. And he and Dr. Hersh also go way back. They were in the same fellowship at Harvard in informatics way back when informatics was new, and then have collaborated many times over the years. So he did record a quick video. He just felt so strongly and felt so bad. So he recorded a video in which he introduces Dr. Hersh personally. So I'm just going to show that video really quickly. Oops. Okay.
Do I need to stop sharing my screen?
No, I think it'll just come up. Yeah, I want you to go ahead and stop sharing so it makes the whole screen.
Yeah, sure, that's fine.
Can everybody see?
Yeah.
Okay.
Hi, I'm Jim Cimino, Director of the Informatics Institute at the University of Alabama, Birmingham, the sponsor of these PowerTalk seminars. And it's my pleasure to welcome today Dr. William Hersh from Oregon Health and Sciences University, where he is Professor and Chair of the Department of Medical Informatics and Clinical Epidemiology. Bill got his bachelor's degree in biology at the University of Illinois in Champaign-Urbana and his MD at the University of Illinois at Chicago. He then did internship and residency in internal medicine at the University of Illinois Hospital. In 1987, he started a fellowship in medical informatics at Harvard, working at Brigham and Women's Hospital with Bob Greenis at the same time that I was doing a fellowship in medical informatics at Harvard at Massachusetts General with Bob Greenis. And we had occasion to work together on several projects. In 1990, Bill moved to OSU, Oregon Sciences University, now Oregon Health and Sciences University. And he has become world famous in his research in information retrieval related to health information and health literature, and also a strong proponent of informatics education. And he was the founder of the American Medical Informatics Association's 10 by 10 educational project. He is board certified in clinical informatics, and he is widely published. He has chapters in several editions of Ted Shortliff's informatics textbook on information retrieval. And as co-editor of that book, I can say we were always very glad to have Bill's contribution, partly because he was always the first one to submit his chapter. He would submit them so early that by the time the book was ready for publication, we would have to ask him to submit an update to the chapters, and he always did that very gladly and promptly. And I was also glad because he has his own book on information retrieval and his own book on health informatics, sort of in competition in some ways with Ted Shortliff's book. So I was always very happy to have his contributions. Bill is a fellow of the American College of Physicians and the American College of Medical Informatics and the American Medical Informatics Association, where I think he is a founding member. If not, he was almost a founding member. He was on the board of directors for AMIA and also secretary of the board. And most recently, he's become an inaugural member or fellow of the International Academy of Health Sciences Informatics, where he's currently the president. He and I have several papers together. First one was 1995. It was the Canon Group, which was a group of informaticians working on foundational representations for medical terminologies. And then he was a lead author for two papers by a wide number of authors, me included, on caveats for the reuse of electronic health record information for research purposes. And if you are in that game, I highly recommend you check out those two papers because they have a lot of important lessons on what to look out for and how to address the shortcomings. So Bill has kindly agreed to speak today at our opening seminar this semester, and his title of his talk is Translational Artificial Intelligence: The Need to Translate from Basic Science to Clinical Value. And I'm very glad to have him here. I'm very sorry that I won't hear this live because I have another engagement. But I look forward to hearing the recording. Bill's talks are always very easygoing, you'll find, very easy to follow. You'll say, oh, I kind of know this stuff. And before you know it, he's tricked you into learning all kinds of new information that you'll then retain forever. So welcome, Bill. Again, very sorry that I can't be there in person to welcome you, but I know you'll do a great job, and I look forward to seeing you at AMIA in November.
All right, Jim, thanks for that wonderful introduction. And Bill, you can start sharing your screen again, and take it away.
Great. Thank you, Jim, virtually. We do go way back. Jim was actually a year ahead of me in the NLM fellowship, so he was one of the kind of senior people, and there's a number of others, you know, who are now old timers in the field who were fellows under Bob Greenis and Octo Barnett back then. So it's great to get that introduction, and I completely understand him not being able to be here. You know, those issues arise with our busy lives. So thank you, Amy and others, for inviting me to give this talk. It is an update, actually, of a talk I gave not too long ago, but I think it's a real important topic. I think, as you know, there's a lot of enthusiasm around AI, machine learning, and there should be, but we also, though, and I think this is where we bring in our informatics perspective, is that there needs to be clinical value. It needs to be beneficial to either patients or healthcare or research or whatever. So basically, this talk is in 3 parts. Actually, as Jim says, probably some of you, many of you will, some of the things I talked about in the beginning part, the kind of promise and some of the early results will be familiar to you. I'm also going to actually spend some time on a systematic review that was done, which I think is really good because it kind of gives us a perspective on where we are. And then, of course, I would be remiss if I didn't talk about my own research, and so the third part of the talk will focus on that. I should mention, and I'll be happy to paste the link in the chat since I can't easily do that right now, that I've made, as I always do when I give talks, a PDF of the slides and all the references that I cite. It's on my website. I actually tweeted it in response to the Twitter announcement of my talk, so you can find it there. There's you can go to my website and you can get these slides. Maybe I'll start with a kind of 1-slide history of AI and machine learning in medicine. Yeah, obviously, there's a huge history. I don't know that 1 slide can do it justice, but I think there are some high points that we need to know, especially for some of you who are newer to the field. I came into the informatics field at the end of the kind of first era of AI, when people like Ted Shortliff, the systems they had developed really didn't quite cut it. I mean, it was great research, it was great work. The technology of the time was much more primitive than we have now, that we're not the kinds of data sources. And so that initial foray into AI really didn't accomplish a whole lot. But AI has been a major activity in clinical informatics really since the beginning, back in the 1960s. And so the first generation of AI was in the 20th century. And the goal there was to try to create these handcrafted knowledge bases and then systems that would operate on those knowledge bases. But as I just said, the computers lack the data, they lack the processing power, they lack the GUIs, and they lack the internet. And so really never had much of an impact in medicine, although we learned a lot, and some of those lessons are actually still important today. This led to the so-called AI winter, which was about when I entered the field in the late 1980s, and led me to kind of focus on information retrieval as opposed to AI, and actually people like Jim Semino to focus on terminology. So we kind of forgot about AI. We stopped even using the term. And then all of a sudden, probably within the last decade or so, we've seen this resurgence, driven mainly by advances in machine learning, deep learning, neural networks, those things. And of course, we have plentiful data, we have plentiful computer power, we've got networks. So it's a whole different world now, and we can do a lot more. There are, you may have your favorite overviews, the book by Eric Topol, the report by the National Academy of Medicine, a review article earlier this year by Raj Prakar are good overviews. But despite all what we've done in AI and machine learning in modern times, there's still modest impact. I had a routine visit with my primary care physician, and she actually does a great job of integrating the clinical visit with the EHR, but there really wasn't any machine learning taking place in the visit that I saw my physician for the other day. So let me talk a little bit about the promise and some of the early results. As you all know, probably the most success has been achieved in imaging. There are great systems that have been developed that can diagnose diabetic retinopathy, the histology of cancer and metastases and pathology slides, chest X-ray findings like tuberculosis and pneumonia, and skin cancer prediction. And so there are systems that work in the lab, I put that in quotes because they're not in general practice yet, that are quite accurate as physicians in diagnosing these conditions. And there's been a systematic review, not the one I'll talk about, but one that looked in 2019 at all the existing imaging applications. And there was a state-of-the-art paper last year from Estiva, who's been one of the leaders in this area. There's other forms of pattern recognition that have been quite effective. Age and sex determination, by actually looking into the retina, things that we as humans can't see, but predicting age, predicting biological sex, interpreting cardiac arrhythmias, interpreting EKGs better than conventional algorithms, detecting hyperkalemia, an important complication that we as physicians need to be aware of, and early diagnosis of things like congestive heart failure. Also sounds, detecting pathological breath sounds has also been something that's been accomplished. I won't go into every last study here, but also taking this, another area has been clinical prediction, predicting length of stay, complications, readmissions, diagnoses. Actually, I meant to say the last slide had age and sex determination from EKGs, but also from retinal images has been another one. And also applying in areas such as COVID-19. These predictive models are very accurate, usually using machine learning, oftentimes deep learning, and clearly there's a lot of potential for impacting clinical practice. Another important area has been assisting humans. We know that one of the biggest downsides of electronic health records has been the time, the way they slow down physicians, nurses, and others entering data. So there are both research systems and some of the commercial companies like Dragon, the Nuance, the company that's now been acquired by Microsoft in part for its medical abilities, assisting pathologists, learning clinical alerts, assisting dermatologists. So another big potential area for machine learning is helping physicians, everything from taking the dialogue between the patient and physician and turning it into notes in the chart, to assisting pathologists, radiologists, dermatologists, et cetera. More on radiologists, and also chart abstraction, the second top level bullet there, is another thing humans spend a lot of time doing. So helping physicians and others extract information from charts and identifying, trying to identify alerts that are more bothersome than useful in clinical care. So how effective are these interventions? There was a systematic review published last year. It didn't seem to get what I thought would be the prominence that it should have in NPJ Digital Medicine. That's one of the Nature offshoot journals. It's a group from China. I've actually had email contact with them because I actually found a few minor errors in the report. But I think it's really important to do these things. How looking at the entire landscape of AI prediction tools. What is their clinical impact? What is their quality? And particularly when it's done in randomized controlled trials. As those of us who are in medicine know, the highest quality evidence comes from randomized controlled trials, where we actually compare the use of these tools with not using them and seeing what impacts they have on patient outcomes. The review looked at all of the clinical prediction tools, and it grouped them into 3 categories. And by the way, the link is here and it's open access, so it's very easy to get. There's what they call traditional statistical tools, mostly things like regression, linear regression, logistic regression. A second category of machine learning, which includes all aspects of machine learning except deep learning, because deep learning and neural networks are not the only kind of machine learning. So other kinds of machine learning and then deep learning. Interestingly enough, and not surprisingly, if you know about these kinds of tools, the traditional statistical and machine learning tools focused on the whole spectrum of activities, assisting treatment decisions, assisting diagnosis and risk stratification. Whereas the deep learning, at least the studies that have been done with randomized controlled trials, focus on assistive diagnosis and really mainly imaging. And yeah, that's, as I said a few slides back, that's the area where where machine learning, deep learning has had the most effect, is with imaging. So this is a figure that you see in all systematic reviews. Maybe some of you have done systematic reviews. I actually have done a couple over the years in areas like telemedicine and health information exchange. But you do this very broad literature search, and then you winnow it down to things like, if you're doing a study of patient impact, things like randomized controlled trials. You could do the same thing on hyperlipidemia drugs. In fact, a colleague of mine down the hall in my office is that he's done a number of systematic reviews. So the methodology is the same. So, you know, there's 26,000 they identified. They have a very broad search strategy, articles related to predictive tools, AI, machine learning, and then they winnow it down to 65 randomized controlled trials from 63 articles, and also some relevant observational validation studies. So we go from this huge corpus of literature down to 65 trials, which actually, if you think about it, is not a lot. I mean, machine learning and AI have so many applications in medicine, yet only as of 2021, 65 trials had been done. And so they looked at these trials. That you may not be able to read well the table that I've copied and pasted from the article, but you can go look at the article itself. They looked at the characteristics of these trials. 3/5 of them had positive results. That's, put another way, 2/5 of them had negative results. They did not show clinical benefit. The trials were done, not surprisingly, on a variety of different disease categories. They lumped them into cancer, other chronic disease, acute disease, and primary care. The most studies have been done with the traditional statistical tools like regression and things, probably a function of time, since machine learning is newer and deep learning is newer still. So there have been fewer studies. And they looked at the function that these predictive tools did. The most common use was for assisting treatment decisions, followed by assisting diagnosis, followed by risk stratification. One of the things that comes up when you do a systematic review is, part of the review is actually looking for bias in studies. Many of these studies had issues with what the systematic review people call risk of bias, meaning that their are methodological attributes of these studies that potentially that are not as good as they should be, and they introduce the potential that the outcome could be biased. So for example, 1/3 of these studies had no sample size estimation. 3/4 of them had no masking. Now masking or blinding can be challenging in some studies, things like, for example, a randomized controlled trial of surgery. You can't blind the patient or the surgeon to the surgical procedure. But these were open label studies, so the people using the systems knew that they were using them, which maybe can't be avoided, but it is a potential risk of bias. Another thing, the majority of these studies didn't use there's a reporting instrument called Consort that should be used with randomized controlled trials. Over half did not use an intent to treat analysis, where you do the analysis based on the group that the patient is randomized into, as opposed to if they may cross over, or provide the study protocol beyond what was described in the methods section of the paper. So one thing to remember, going back up to the top of the slide, that sometimes people say, well, there were 60% positive results and 40% negative results, so that's good or not good. The number of positive studies isn't necessarily an indication of superiority of one method over another. Another table from the paper just gets into a little bit more detail. It reiterates, so for example, what kind of data do these trials use? So again, not surprisingly, the traditional statistical and machine learning systems use clinical quantitative data, so structured data out of the EHR. The deep learning studies tend to use images. Interestingly enough, very few of these studies make use of the text. Many of us, myself included, are very interested in using the text in progress notes, discharge summaries, et cetera. I think just 1 or 2 of these studies actually did that. The disease categories were varied. The deep learning studies were focused mostly on cancer and mostly on cancer imaging. The tool function varied by the type of tool used. And the results were mixed. They were actually more positive for machine learning and deep learning. But again, the numbers of studies is small, and we can't just go by the number of positive studies. Some other interesting observations, the number of studies increasing over the years, which is a good thing. And the rates of positivity were actually higher for deep learning over machine learning over the traditional statistical methods. So although again, the number of studies is small, this gets into the kind of risk of bias that we see. And we really need to hold our community to good randomized controlled trial methods if we're going to publish these randomized controlled trials and make a case that we should be using this AI tool or that AI tool in clinical practice. And some of the things I've already mentioned, the suboptimal use of the consort statement, which actually has been updated. There's a consort AI statement that is specific to AI, sample size pre-estimation, randomization, intent to treat analysis, and so forth. If you're interested in those things, you can delve into this paper. I just wanted, so the paper, again, breaks the tools down. I think it's very interesting to look. This is a table from the paper. It's not particularly easy to read, which is actually one of my complaints about this paper. But as of 2021, there were only 11 randomized controlled trials that had been done looking at deep learning tools, and 9 of them focused on assisting endoscopy. Those of you who are not clinicians, endoscopy is where we stick those flexible tubes and various orifices in the body. These were mostly GI endoscopy, so either upper or lower GI endoscopy, and looking for things like, helping detect polyps that could be cancerous or could indicate a risk of cancer. All of those studies actually had positive results. And many of them actually come from just, I think it's 2 or maybe 3 research groups in China who do really good work, but it's not, we don't know how generalizable that is. And then the 2 other studies, one that was used for ambulance triage, and then out of hospital cardiac arrest, and then the other one is in ophthalmology. So looking at images inside the eye, and that study was negative too. So what this means, it doesn't mean that deep learning has no role in medicine, but it means that we need to find the proper way to use deep learning in medicine and actually show that we can have positive benefits. So what conclusions do I have about this review? I think AI predictive tools do still have great promise, but we don't yet have the comprehensive evidence that we need to advocate for their use. So the number of clinical trials assessing clinical benefit of AI is still small. The majority of the trials published so far have intermediate or high risk of bias. It doesn't mean that they're biased, but there were not methods used that would minimize the bias, and that the trials of deep learning methods, despite all the what I call basic science studies in use of deep learning, are mostly focused on endoscopic procedures, which is good for endoscopic procedures, but we need to look at more areas. There are a few other kind of minor issues with the review. There was, and actually, this is what I was mainly emailing with the authors about. They actually had a missing column in the paper, in the print, in the journal PDF paper of the deep learning interventions, which makes it very difficult to interpret that table. They did send me a spreadsheet that had that table in it. I don't know if they ever updated the paper. And then there was a study that wasn't there of a deep learning intervention, a randomized controlled trial, which highlights the challenge with static systematic reviews. The day you publish them, they become out of date because new trials are coming down the pike. And I encourage these authors to keep their systematic review up to date, which groups like the Cochrane collaboration do. If you publish a systematic review with them, you actually also agree to keep it up to date. Some of the data is difficult to use. There's a supplementary table 4 that has all the machine learning interventions, and it's a PDF that's spread across pages. It's just a very unusable thing. They did send me a spreadsheet with that, and I encourage them to really publish that as their supplementary data and not a PDF printout of a table. And then there was another study that I thought should be there that wasn't, but it may have been a traditional statistical, although they didn't publish a study. They didn't publish a table that had their traditional statistical interventions. So, I mean, all in all, a very good review. I hope they keep it up to date because we, as informaticians who are going to be implementing these AI and machine learning systems in clinical settings, need to know how to need to know whether these things actually work or not. When clinical colleagues come to us and say, "I want to implement this tool in the GI clinic," well, is there benefit and so forth. Okay, so then, as I said, the third part of my talk, and I tried to leave some time for questions and discussion. Always interested to hear what the audience has to say. Applying, so this is my work. It doesn't actually involve a lot of machine learning. You could debate whether or not it's AI, but anyways, it is pertinent to this topic, and so I thought I would tell you a little bit about that. So, as with all informatics research, we need a good one or more good clinical use cases. There's a lot of interest these days in cohort discovery, so identifying cohorts of patients, whether you want to follow them or recruit them for clinical trials. There's a lot of tools that have been developed that are good, although not great. And then another focus, which actually it resulted from people coming to my group at OHSU, trying to diagnose people who might have rare diseases, which often go undiagnosed. or go undiagnosed for a long time. And because I work in information retrieval, part of the process applies information retrieval to this data. And Jim Semino mentioned my book when it was published a couple of years ago. I took this picture, or actually my wife took this picture of me holding both print and electronic versions, which I don't know if your institution subscribes to Springer Link, but if it does, you can go download this and all the other Springer Informatics books. Okay, then, so what data did we use for these use cases? OHSU, like I know the UAB Informatics Institute, has a research data warehouse of data that comes out of our, in the case of OHSU, EPIC system. This is fully identifiable data. This is not de-identified data, because our belief is that for this kind of research, we really need the real thing, which then creates a problem in that we can't share our data outside the institution. But our IRB is comfortable with us doing this kind of research on fully identifiable data in our data warehouse, even potentially my record, because I'm a patient at OHSU. We try to minimize the research staff, though, going in and looking at the patient data, although we have the IRB approval to do so, and sometimes we do need to do so. This work has been funded by grants, mainly an R01 from the National Library of Medicine. But for these rare diseases, there's actually a lot of pharmaceutical companies that are interested in it because they've developed these drugs that are only applicable to a small number of patients because they're rare diseases. And so they're interested in potentially diagnosing more patients. This is not work that I do by myself. Some of you may know some of the people listed here. Steve Bedrick and Aaron Cohen are informatics faculty members at OHSU. Steve Chamberlain is a postdoc in our group. And Tom DeLowry is a neighbor of mine who actually is OHSU's expert in acute intermittent porphyria, which is the disease that we were funded by Al Nyland to study. And I'll talk a little bit about that study, including, spoiler alert, a negative result from the outcome, but we'll get into that. I'm only going to talk briefly about the cohort discovery work because that's a work in progress. We actually had an initial R01 and we had some initial studies, but we spent 5 years learning of a number of challenges of this kind of work, trying to do work with large quantities of clinical data. But we obtained a collection of 100,000 patient records, the complete records out of the research data warehouse, which came from the original EPIC data at OHSU. And we published on the results, on the methods and the results of the initial work with that data. We then got the R01 renewed. It was original collaboration with Mayo Clinic and OHSU, and we've added UT Houston, and we're updating our data systems and methods. One of the complaints of the reviewers of the renewal was that, how can you generalize your results across institutions when you can't copy, you can't share your data. We can't share our data. There's no way that any of our institutions would let us. There's no way OHSU would let me send 100,000 patient records to Mayo or UT Houston or UAB or whoever. But so our response, which the reviewers seem to buy, is that we will standardize everything else. And of course, there's a nice standard now for the format, namely OMOP, the Observational Medical Outcomes Partnership, which allows, which gets the clinical data into a standardized format and standardize the terminology and so forth. And of course, any tools we develop will open source and we'll describe our methods. So we're basically, we'll be sharing everything except the actual data. And then institutions, if they want to do this kind of work, will need to load their own data and then develop build their own models and see what results they get. But there are major challenges to this kind of work. One is that even though we're moving towards more standardized EHR data, the records are still heterogeneous. A problem that we just recently have come upon is that different instances of epics use different, and the institutions that run them use different names for notes. You know, part of our methods involve processing the text in notes. There actually is a standardized ontology for notes in LOINC that we're going to use, but we'll have to map what our systems use. But you know, this just goes to show how data standardization and interoperability are important, but also challenging to achieve. And then the whole issue of privacy concerns. My earlier work in information retrieval looked at journal articles, Medline abstracts, textbooks. There are copyright issues, but it's not nearly the kind of privacy concerns that you have with patient data. And I think that's always going to be a challenge for this kind of work. So that's all I'm going to say for that. And I'll finish up next several slides talking about rare disease detection. This was a fun project. We had hoped, well, it's really not over yet. We're actually even talking about expanding the study. So let me talk first about rare diseases. There's probably more than 1,200. It depends on how you define rare diseases. But there are many rare diseases out there that affect small numbers of patients. The usual standard definition is less than 1 in 200,000. And many of these diseases are underdiagnosed. There's a website, rarediseases.org. Many people have written about it, including Dr. Melissa Handel, who's now at the University of Colorado. And the characteristics of these diseases that primary care physicians don't really think of them, which they really shouldn't, because they're dealing with common diseases like diabetes and hypertension and all those things us general internists, or in my case, former general internists, look at. So there's a disease, acute intermittent porphyria. If you're a physician, you probably learned about this in medical school. I certainly did. You know, I learned the genetic pathway and where the mutations are, you know, and then I promptly forgot about it. I've never seen a case of acute intermittent porphyria, which is also sometimes called acute hepatic porphyria. But there's a metabolic pathway to synthesize the heme protein, which goes into hemoglobin, which goes into your red blood cells, which takes oxygen when you breathe in and distributes it to the tissues. So heme is an important protein in human, actually all animal physiology. And there's a pathway, and the enzymes, there are enzymes or proteins in the pathway that assemble the heme molecule. And when there's a mutation in the gene for the enzyme protein, you don't get the proper production of the heme molecule. And for those of you who know genetics, there's variable penetrance, which means you can actually have the mutation, but it may not cause any symptoms in you. The incidence of acute intermittent porphyria is 1 per 100,000 in the population generally. And it's another one of these diseases that often go undiagnosed for a long time. But the symptoms are significant. And if you've ever taken care of a patient with acute intermittent porphyria, they have significant morbidity. They have so-called neurovisceral symptoms that include abdominal pain, nausea and vomiting, and psychiatric changes. These are all fairly common symptoms that are present in so many diseases. So that's another reason why people don't think of acute intermittent porphyria, although it is rare. The people who have the symptoms are acute and intermittent, and people get really debilitated by this condition. Ironically, the disease is diagnosed by a relatively inexpensive urine test. There's a chemical porphobilinogen, which since the heme pathway gets backed up because of the genetic defect, this porphobilinogen builds up and then spills into the urine. So it's a $70 test to actually diagnose it. You can also do the genetic test, although again, with the variable penetrance, just because you have the gene doesn't mean you actually have the disease. And then what really changed things, and what gives companies like Alnylam the incentive to try to find more patients who have this, is that there's a new highly effective and as with most diseases, most treatments for rare diseases, highly expensive treatment. There's an RNA silencing molecule called givosiran There was a study, randomized controlled trial in the New England Journal of Medicine. It really changes the quality of life for these patients. So yes, it's an expensive drug, but if you do have acute intermittent porphyria, this drug can potentially change your life. So it's worth trying to find more patients with it. And we can debate the economics of drug pricing in a different discussion, but this just goes to show that these drugs can be very effective. So the question that motivated Al Nylam to come to our group is, can we detect rare diseases earlier using population-based techniques, basically using EHR data? So we took the data set we had from the previous study, the cohort discovery one, we added 100,000 more patients. So now we have 200,000 patients. We also wanted to make sure that we would have all patients who might have acute intermittent porphyria. So we found another 5,571 patients who had the string PORPH somewhere in their record. So that gave us about 205,000 patients. So then, and we thought we would develop a machine learning algorithm. And this is mainly the work of Aaron Cohen, who some of you from AMIA know who Aaron is. And we found, actually, so of these 205,000 patients, 47 had the ICD-10 CM code E80.21, which is acute intermittent porphyria. And then we manually reviewed. We actually had a medical student who wanted to work on a project, as all of our medical students do at OHSU. And her task was to review these 47 patients and see how many of them truly had acute intermittent porphyria. And we came up with 30. Now, for those of you who know machine learning, this is a highly imbalanced set of 205,000 records, only 30 of which are positive. But all of the rest of the patients were the negative training cases, and we applied the machine learning. And if someone asks me a real detailed question about machine learning, I may have to refer you to Aaron Cohen. I've certainly learned about it, but he's the real expert in machine learning. And so we parsed the record into features and sorted them by frequency of how they appeared. And then we did a univariate feature analysis. So we looked at features that would go into our machine learning model. Now, there are some other things that we wanted to keep out of the model. So we didn't just do this kind of blindly. So for example, the word DeLowry, I mentioned our expert, shows up because he's the provider at OHSU that follows all these patients. We got the drug cimetidine. It actually turns out that there actually was a case report of cimetidine providing benefit in acute intermittent porphyria, but we wanted to choose features or terms that were generic and would be used in other places. So we trained on the full data set. Aaron tried a bunch of different machine learning methods, and he found to get the best predictive performance with support vector machines using radial basis function kernels. And then we applied the training model back to the full data set to rank patients by the margin distance, by how close they were to the positive cases. So this basically gave us all of the other patients that we could look at to see if they may indeed have acute intermittent porphyria and potentially test them. We identified, if you look in the lower, so we looked at all the patients. We focused on the ones that did not have any mention of porp. There were also those that did have mention. We did some additional work with them, but our goal was to try to find patients who may have acute intermittent porphyria that had never been considered. And we looked at 100 patients. We probably could have kept going down the list, but time was limited. And we actually identified 22 patients who had the classic symptoms of acute intermittent porphyria, but had never been even tested, had never even been considered. So those are the patients that I'll get to on the next slide. We did a clinical validation study. In terms of the other patients who had the symptoms, there were patients with things like cancer pain, psychiatric diagnoses, and so forth that did have mention of PORF. Well, actually, even some of the ones in the without mention of PORF, actually, those were some of the diagnoses. So other kinds of pain, other kinds of psychiatric diagnosis. So we actually decided that kind of, since I talk about this, I have to put my money where my mouth is, or whatever that phrase is, to do a clinical study to actually see if we could see if any of these patients actually had acute intermittent porphyria, and it would actually benefit them because they would be candidates for givosiran, that new drug, which is actually covered by most insurance and Medicare. So we set out to do the clinical study. Unfortunately, we set out to do the clinical study in late 2020. That was when the previous paper that Aaron Cohen and I had published. It took us a long time to convince our IRB to let us do this. And there also happened to be a COVID-19 pandemic, and this was before there were vaccines for COVID-19. So we negotiated a lot with the IRB. They appropriately so wanted us to not just go to the patient, but to go to their primary care or other physician first. And then if the physician approved, then we could contact the patient. But our goal was to identify those 22 patients, the 4 likely, 18 possibly, and test them to see if they may have acute intermittent porphyria. They had the symptomatology, but no one had thought about it. This slide is a little busy, but basically, so we have those 22 patients. Turned out that 6 of them were either deceased or had moved away from Oregon, so we couldn't figure out how to contact them. 12 were receiving their care at OHSU. 4 had transferred their care outside OHSU. So, and then within OHSU, some had primary care physicians, others were seen by specialists within OHSU. And so we asked the clinicians whether they would let us contact the patient. 3 of the physicians said no, the rest said yes. And so then we contacted the patients. We tried email, we tried calling them. This was at the height of the pandemic. But we got through to, I think, about like 15 or so, and actually got more than 7 people who agreed to come in and give a urine specimen. But the other ones did not. And so we tested 7 patients with urine porphyrobilinogen. It's not, even though it's an inexpensive test, it's not an easy one to do because the specimen has to be put on ice as soon as it's collected. Otherwise, the porphyrobilinogen disintegrates. So they did need to come in. I mean, potentially we could have sent people out, but we didn't really have the resources to do that. And 7 people did. And needless to say, the 7 who came in had normal urine porphyrobilinogen. So this is a negative study, but can always put a positive spin on things. I think this is important. I think it's important to do these studies in the first place because we have a great algorithm, and it may be that if we test more people, which we're hoping to do in the longer run, we may find someone, and we may be able to get them on that drug that's beneficial. Another challenge is for those of us who are informatics researchers who are not directly caring for these patients, how do we care for them? The 2-step approval process, which is a good thing because it does involve the physician, and many of them were helpful in helping us contact their patients, but it does add complexity. Another issue is that rare diseases are rare. So we already had a small number of people diagnosed, and the question is, when the incidence is 2 per 100,000 people, where we're going to find more people who indeed have the condition. And then this doesn't apply to our study, but testing may be expensive or even harmful. If there needs to be a biopsy or even a blood test is not really harmful, but it's more expensive, it's invasive, and so forth. But still, we did identify people who were at least candidates for testing for urine porphyrobilinogen, but we didn't identify any positive cases. So what do we conclude? And then I'll stop sharing and happy to have some discussion. So as in all of medicine, we do things in the basic science labs, whether it's machine learning or a tissue culture study or whatever, but they must achieve clinical validation. And many machine learning models, our literature, including our informatics journal, is filled with papers that have achieved what I would call basic science success. And now we need to demonstrate the clinical value. And then, since I know many in the audience are informatics researchers or students or fellows who aspire to become informatics researchers, I think this is probably one of the most opportune areas for informatics going forward, of doing these clinical studies, collaborating with clinicians, and seeing how our machine learning and AI tools can make a difference. So I want to thank you all. Again, you can get these slides, which has my email, my website, my blog, my Twitter feed, and I'm happy to have any further discussion. So I'm going to stop sharing here, and I'm happy to answer any questions. It looks like there are questions in the chat. Let me just ask Amy if she would prefer me to start there.
Yeah, we've had some really great questions. Thank you, everybody, for posting them. You can either read them or I could read them for you.
Yeah.
It's like you have access to them.
Sure. No, I'll go down the list.
Really great questions. Thank you, everybody.
Yeah. This is always the best part of giving a talk. I actually was hoping I would get through a little faster, but anyways, okay. So there's the link to the PDF that Amy posted. So from Rahul Sharma, how do we do a standardized tool trial? Is there any authority I can connect to? I'm not actually sure what you mean by standardized tool.
I think he means that it's so easy to do a drug trial. There's this clear channel for how you do that.
Right.
How would we do the same with tools?
Sure, gotcha. Okay, yeah. There are, I mentioned the consort statement, which is if you do like a drug trial, you're supposed to make sure your paper covers the data elements in the consort statement. And that statement is, there's now a consort AI statement that does the same thing with an AI perspective. Randomized control trials are not easy to do. I mean, you have to get IRB approval, you have to go out and recruit patients, and you need to randomize them and things like that. It's not easy. I mean, certainly the best thing to do is to collaborate with someone who has experience in doing those kinds of trials. I would actually probably your institute in terms of who you should connect to. I imagine there's people that can help you. How deep learning can help your research? Well, if I'm understanding this question correctly, how can deep learning help medicine? And I think it's important to do the basic science studies of machine learning to show that your learning algorithm can diagnose diabetic retinopathy or pneumonia, or predict poor patient outcomes, things like that. And you need to do that initially in a kind of basic science lab kind of way. But then, we need, can you actually take a machine learning algorithm that can read chest x-rays? And does it actually work in a clinical setting where the world is not quite as perfect as in basic science kinds of studies with clean data sets and all that? So they have to, we need to test them further. Another issue with imaging studies is there are lots of different imaging equipment, different vendors and so forth, which the imaging people admit that we need to deal with, because different vendors' imaging equipment might take pictures slightly differently. I'll keep going.
There was another question, I think, about that one. That
okay, sure.
Okay, yeah.
I'm happy to jump to it, or do you just want me to keep going down the list?
I'll just read it out. There's certain EHR companies hinder help in machine learning. Could you give an example where an EHR happened to be better than others?
Yeah, no, that's a very important question. I think and I mean, in some ways it shouldn't even matter because, but it does, and I'll address that in a second. But, what we're interested in is the data, of course, then the EHR is the vehicle to collect the data. I think the EHR companies are going to have to help us with this. And I mean, I've seen positive signs from some of the vendors, including Epic, that they're interested. But what's really important are things like data standardization. So things like FHIR and the new regulations from the 21st Century Cures rule. So I don't know that any one EHR is better than another. I don't know that anyone's actually looked at that, but it is important that we partner with EHR vendors and they partner with us, and that they recognize it's important for their tools to be able to assist with this kind of research. Next question, Ida. Oh, Ida, great to see you. I see your still picture on the top of my screen. There you are. Did the negative results lead you to review and or revise the algorithm? They haven't yet, but certainly that's a question because people say, well, why didn't you use a deep learning algorithm? Aaron actually tried deep learning, but he actually got better results with the support vector machine. Now, there's been advances in deep learning since we did that work. So we probably need to go back. We're actually hoping the vendor Alnylam is actually interested potentially in additional research, but we would obviously need the resources to do it. But certainly, I think we probably need to go back to the beginning and look at new algorithms. Update our data set, and then potentially, and we probably need to test more patients than just the 7 who we convinced to show up. Does that answer your question?
Well, I was thinking more if there were characteristics of the 7 or potentially more that you might get in the future that would lead you to change the algorithm.
Yeah.
Because they were negative in terms of the disease, right? So you've got an example of negative.
Yeah. Right. Well, yeah. So we don't have a positive in the sense that we diagnose them. But I think we do need to look at other algorithms. We probably need to look at other data sets. Where there's actually another institution that we're potentially going to collaborate with to look at their data. So yeah, I don't think this is the final word. And in fact, one of the issues with publishing that small paper, there were reviewers who didn't want to publish that paper, but I thought it was important to publish it to just show that we need to do this kind of work. I don't think this is the end of the story, though, and that's why we published the paper as a brief communication and not as a full-on paper. But yeah, I think there's a lot more work to be done, and I don't think what we did is the end of the story. Yeah. So I'll let's see.
Also, just to do a check on time, that we're at time, so I don't know if you're able to stick around or if anybody else is able to stick around, but feel free to leave if you need to, because we know that people have lots of things going on.
Sure.
Friday before Labor Day.
I can stick around another maybe 5 or 10 minutes. I'm actually giving another talk today to medical students about informatics. It's a talk I give each year. But yeah, I can stick around. Why don't you triage the questions to me, and then you just read them out, and I'll try to answer them.
Okay. Do the negative results mean that the learning tools were not beneficial, or they have negative effect on humans? So I think whether that just means like neutral, like you didn't find anything, or there was some kind of harm, I think is what the question is.
Yeah, well, I don't think there was any harm. Many of these patients that have the symptoms of acute intermittent porphyria have these symptoms for a prolonged time, and they're undiagnosed, and we didn't find any that were diagnosable. So we didn't do. I can't imagine anything we did here was particularly harmful. I suppose we made people venture out of their homes before there was a COVID vaccine, but we had a protocol that where we obtained consent and we explained to patients exactly what we were doing. And so I don't think there's any harm here. Now, if this were a different disease and might require, well, a blood test, is not really that harmful, but you do have to get stuck, or maybe something like a biopsy, then you're potentially getting into harm. We didn't have that particular problem here. And I don't think we gave any patients any kind of false hope or anything, but those things need to be considered as we do these kinds of clinical trials.
What is the best way to, how would you minimize bias with a deep learning model? Since you talked about, look for sources of bias, how do we minimize bias?
Well, yeah, there's all kinds of bias. I mean, first of all, there's bias in data, there's bias in algorithms. Those are big issues for us in the informatics world. There's also the bias that systematic review was looking at was just clinical trials methodology that wasn't as good as it could be. When you do a big multi-center randomized controlled trial and you adhere to the consort statement, you address everything like concealment of allocation, so the researchers don't know which groups the patients are randomized to. They may find out, they will find out eventually, but that they don't influence the process. Intent to treat analysis, so people get analyzed in the groups in which they were randomized into, and it sometimes, especially like with drug trials, or with like surgery trials, people cross over. So all of the kinds of things that are important methodologically in randomized controlled trials, we should be doing that too when we do these kinds of clinical trials. The study that I did wasn't really a randomized controlled trial. It was just really kind of more of a clinical validation study. But for other kinds of things, for example, for predicting deterioration in the hospital, you need to validate your algorithm, but you also need to show that algorithm actually can identify those people and give the clinical team an opportunity to make a difference. Otherwise, you're just predicting something that you can't do anything about.
What is the best way to compare AI and machine learning with traditional methods like regression? Can we compare results and draw conclusions based on this? And also, he's citing an article, a systematic review shows no performance benefit over logistic regression clinical prediction models.
Well, actually, I don't know that the systematic review shows that. I don't think they actually compared. These clinical studies tended to compare the use of AI versus the non-use of AI. That's what most of the clinical trials did. But getting back to the first question, if we have good data sets, we can compare any kind of machine learning model. Regression, neural networks, deep networks, support vector machines, et cetera. So at the basic science phase, we can compare the different models and we can see which one seems to work best. Then presumably we would take the one that works best and then advance that into a clinical trial where we would put it as part of the care of the patient, and the patients would be randomized to care with that tool versus care without that tool, or with some other kind of best known practice kind of tool. So you can, with good data sets, you can compare different statistical, machine learning, other kinds of models. But then when you actually do a clinical trial, you want to see if it actually leads to some aspect of better patient care, better patient outcomes, or lower costs, or whatever.
Just there was a quick comment about that previous question and that paper, that logistic regression is a machine learning model. So
yes,
it's not an either or necessarily.
Right. Yeah. So, machine learning is, the broad kind of, as the term says, machines that learn, and machines learn from, when they calculate logistic regression, they learn, when you build a deep learning model, and everything in between.
All right, that's all the questions that we have. Any final questions? You can feel free to unmute yourself if you have a question or a comment. Oh yeah, get kudos, people clapping, and oh well, seeing none, hearing none, then thank you everybody for attending, and thank you for presenting this great presentation, very relevant and of interest across a broad cross-section of our community here, informatics, translational, clinical, technology. So well, have a good Labor Day weekend, everybody, and keep doing all these wonderful things in research. Thanks very much,
And-
Bill.
And I will look forward to seeing some of you at the AMIA meeting in November. So,
Oh, yep. Yeah, we all have friendly faces here, and yeah, see everybody there. Probably that's
Okay.
how we network there. All right.
yeah.
Take care,
Great to
everybody.
see everyone.
Bye-bye. And
Bye-bye.
be sure to come for future sessions. Next week, we have one at 9 and 10. We have like a double header next week. All right.
Thanks,
Bye,
Dr.
everybody.
Hersh.
Yeah. Bye-bye.
Bye.